I got so much motherfucking design on right now.
Break it down, bring it back, make it clap. I got 3 old thoughts, new block, where that thing. Bitches on my line, and you know they come straight.
Hello everyone, so today we are from group 5 gonna present about copper and manganese in plant and soil. Copper is essential to the growth of plants and also classified as a micronutrient. Among other things, it plays a part in several enzyme processes and is key to the formation of chlorophyll. It has a role in various important processes in plants. For example, it is a structural element in numerous proteins. Not only that, copper also activates some enzymes in plants which are involved in lignin synthesis and also essential in several enzyme systems. It also requires the process of photosynthesis, is essential in plant respiration, and also assists in the plant metabolism of carbohydrates and proteins. Moreover, it also participates in cell wall metabolism. Copper also serves to intensify flavor and color in vegetables and color in flowers. Copper availability in soil and uptake by plants. Copper tends to easily bind to organic matter. Therefore, soil organic matter reduces copper availability to plants, and despite their low pH, copper deficiency might occur on acidic soils if the organic matter content of the soil is high. For manganese. Manganese is an important plant micronutrient and is required by the plants in the second greatest quantity compared to the iron. It is also important micronutrient for plant growth and development and sustain metabolic roles within different plant cell compartments. Like any other element, it can have a limiting factor on plant growth if it is deficient or toxic in plant tissue. Function of manganese in plants. Manganese is used in plants as a major contributor to various biological systems, including photosynthesis, respiration, and also nitrogen assimilation. Manganese is also involved in pollen germination, pollen tube growth, root cell elongation, and also resistance to the root pathogens. It also participates in chlorophyll production. Not only that, manganese also has a role in enhances starch production. Manganese in soil. The reactions of manganese in soil are complex. The two major factors that affect manganese availability are pH and redox conditions. Other factors include soil, organic matter, microbial activity, soil temperature, and also seasonal variations affect its availability to the plants.
I am going to share about the effect of excessive copper in soil. Study shows that excessive copper affects the soil by reducing the level of nitrification, denitrification, and soil respiration that occurs in it. This has an impact on the plants that depend on the product of this process to thrive. Soil ammonia is oxidized to nitrates, NO2, and then to nitrates, NO3, in a two-step conversion involving various types of microorganisms. The conversion of ammonia into nitrates is done by autotrophic bacteria and archaea, and the transformation into nitrates is done by nitrobacteria and nitrospira. Both of these processes are necessary for the eventual release of nitrogen, with plant producing proteins essential for the growth. When copper is in the soil in significant quantities, they can prevent this process and shrink the production rate of net vital. This appears to be worse in coarse and medium texture soil, where nanoparticles prove most toxic to nitrolic bacteria. Next, denitrification. Heterotrophic bacteria also break down nitrates into molecular nitrogen in a process known as denitrification. Abovalent depends on nitrogen to make the cell proteins, where nitrogen-making bacteria are found to be affected by excess copper. Plants that benefit from their nitrogen-making activity may also suffer. Last but not least, soil respiration. When soil microorganisms, they produce carbon dioxide. Carbon dioxide is released into the air, captured by the plant and converted into organic compound through photosynthesis. Plants either use compounds to build their structure or extract energy from them during respiration. Respiring soil organisms are also affected by toxic quantity of copper, as they are forced into degradation by this compound. Less carbon dioxide is released into the air from the soil, a situation that may affect agricultural ecosystem in the medium to long term.
I will present about the copper in plant. Copper play an important role in plant development. Copper also the key component for the chlorophyll because it occurs during the photosynthesis and production for vitamin A. However, it is essential for photosynthesis process where plastocinin is a copper protein that acts an electron carrier in the electron transport chain of photosynthesis. The deficiency and symptom of copper are similar, which is copper is a mobile nutrient and the deficiency symptom appear on the tips of young leaf. Meanwhile, the symptom then extend to the leaf margin. For example, the new germinate leaf will be chlorotic, which means the chlorosis occur between the vein in young leaf. Besides, the fruiting and flowering will inhibit in annual plant. That is the deficiency in copper. In addition, the symptom of copper is the leaf will wilting, the young leaf will become bluish green. Next is the twisted young leaf, and last symptom of copper is the flowering will be delayed. The next point is the toxicity of copper. Copper is essential micronutrient. The excess of copper might be toxic to the plant. It might inhibit the plant growth by causing an oxidative damage to the cell and interfering with the photosynthesis process. When it excess, copper may also replace the magnesium in the chlorophyll molecule and impair the photosynthesis process. In addition to the direct toxicity, the excess of copper may also cause the antagonistic interaction with the other nutrients. Therefore, the excess of copper may result in deficiency of nutrients such as molybdenum and iron. Copper toxicity in plant depends mainly on the plant species and soil condition. The copper toxicity in alkaline soil is less or low likely to occur than acidic soil, which means copper are more occur when in the acidic soil. The toxicity symptoms such as the necrosis, stunting, and the roots will inhibit, and the shoot also will inhibit. The last point of copper is the effect in copper. High copper can make the soil become acidic and peaty. Secondly, if the organic matter are enough to decompose in soil, it can make the copper released to soil abundantly. Next is high copper in soil can make the plant inhibit, especially during the flowering process. And lastly, copper element is essential to plant respiration to metabolize the carbohydrate uptake.
So I will present about the manganese in soil. What does high manganese in soil mean? Manganese toxicity is major limiting factor in acidic soils. Soil conditions that favor accumulation of toxic levels of manganese, soil pH lower than 5.5, lower calcium levels in the soil, lack of oxygen as a result of excessive irrigation, poor soil drainage, soil compaction, and high precipitation. Manganese toxicity can be treated in various ways, such as application of magnesium, application of organic matter, application of lime for soils with low pH, and prevent fluctuations in soil moisture level. Manganese deficiency results in reduced crop yields and quality mainly due to impairment of the photosynthesis process and synthesis of starch. Deficiency symptoms begin as interveinal chlorosis of younger leaves and or necrotic spots. Deficiency occurs mainly in calcareous soils, soils with high pH, soils with high organic matter content, and in poorly rated soils. Excess of iron might also cause manganese deficiency as iron competes with manganese for uptake. In calcareous soils, a foliar application is recommended for correcting manganese deficiency. When growing in soil, excessive quantities of magnesium do not appear quickly. Too much magnesium inhibits the uptake of calcium, and the plant displays general symptoms of an excess of salts, such as stunted growth and dark-colored vegetation. Manganese deficiencies are most often observed on well-drained neutral or calcareous soils. However, other soils may cause manganese deficiencies, particularly as a result of heavy fertilizer usage. It can also be induced on these soils by heavy applications of lime. Manganese deficiency is controlled by using manganese sulfate (MnSO4 0.7H2O) as a soil applicant or a foliage spray. Chelated forms of manganese can also be used as a foliar spray, although this treatment is more expensive.
Manganese is important micronutrient for plant growth, develop, and maintain metabolic roles within cell part. Manganese required in small amount when we want to apply it on plants. Here's the toxicity of manganese if we apply it excessively. Manganese toxicity can trigger oxidative stress and disrupt the photosynthesis. Next, inhibit enzyme activity in plants, and the last one is restrict the uptake of calcium. Dose toxicity, of course, we can determine by symptoms of the plant, which the symptoms of toxicity may cause the plant chlorosis in younger or upper leaves. For example, brown spots surrounded by irregular areas of chlorotic tissues and pale or yellow color at leaf due to insufficient of chlorophyll. Next, the plant growth will stunted, necrosis in the older leaves of vine. Lastly is irregular shape, patches of pale tissue in intervenous zones or crinkled leaf. As for deficiency of manganese in plant, it will possibly cause the chloroplast development at the chlorotic spot, which will reduce the chloroplast development. Second, reduce chlorophyll production, which the plant leaf show unnatural coloration. Next is chlorosis. It presents around vein that cause abnormal dark veins. Lastly, necrosis. For effects, the first one, as we can see here, the picture show that intervenous chlorosis with wide area of green associated with veins. The picture show manganese deficiency in Tung oil tree, Elorites 4D. The source from this picture are from University of Florida. The effect of deficiency of manganese, also the plant parts will reduce in size. Next is necrotic spot, which the spot is in tan or in gray color. Last one is make the plant growth stunted.